Right. So are you all there? Can you hear me? Hello? I just see I
Yes,
have.
ma'am.
Yes. So hello, how are you all? I'm seeing you after a long time now, no? There's a problem with the light today. It's very cloudy here. So are we expecting some more students or good morning everyone? Yes. Are we expecting some more students or do we start with this now? Do I show you it's morning 8 a.m. here? 8:37, right? And I'm calling from my home, and see, this is the outside window from where this light is coming, which is a bit low at this stage. And this is the view. You have a lovely view from here. And these are a lot of pine trees outside my home, and we see a very good sunrise. But today sun is behind the clouds. Can you see sun now? Sun is yeah, it's behind the cloud, behind the tree. Maybe it'll come back in a minute. And on the left side there are a lot of good pine trees here, and the atmosphere is very good, right? So it's very early in the morning here. So I was sitting in front of the window, so light on me is at the mercy of the sun. Right, so two students are already waiting, so let us admit them. And I think 7 is a good number, right? 7 is a reasonably good number. So do we start? I have just woke up, and what is the thing is because you have limitation of everything, and still it was good that I could talk to you before your exam. I was feeling a bit this thing. So do we start now? What we're doing now is we are discussing about your forthcoming exam on 2nd of January. That is bioinformatics and biostatistics. So you want to straight shoot the questions, or how do you want to go about it? Just let me know.
मैम एक क्वेश्चन था वो डिफरेंट टाइप ऑफ ब्लास्ट वो
Different types of sorry blast. Right, there are many types of blast. One is nucleotide blast, blast P for proteins, and there are 8-9 types of blast. Do I share the screen and show you? How will you get this answer?
यस मैम
Yeah. So I share my screen now, and share my screen, and this is the window I want to show you. So share. Can you see my screen now? Please keep interacting. It's a meeting, not a webinar, right? And do you see so many types of blast here? One
Yes,
is
ma'am.
nucleotide blast, where you can just blast nucleotide sequence. This is translated nucleotide. Protein blast X is protein. And this is translated, this is N again nucleotide, but the translated nucleotide. This is protein blast, and there are several new types of blasts which have come. And I had given a lecture at, I remember, at CEC. You can see there also. So, but I think from you, we just expect these 3, 4, 5 types of blasts. Is it all right?
Yes, ma'am.
So you have to write about these type of blasts, and meaning these 3, 4 are very easy to remember, but if you do it a bit more, you will be able to you can make a consolidated answer for this. If you have problems, I can send you that PPT of mine where I talked about all type of blasts. Okay with this? So I stop sharing the screen and come back to you. Now next question. So all of you took microbiology in next semester?
Yes, ma'am.
And you know what is I am teaching here? Next semester I am teaching here, and the paper I am teaching is microbial community analysis. And So this is what I was telling Dr. Anil, is that if you want to do a good project in microbiology, I'll be able to get it done from here, meaning I'll be able to teach you from here and ask you to join in the same class, maybe, if it is possible, or otherwise we'll figure out what ways we must have to do this, right? So that is one thing. And if you have any other questions, you can ask me quickly, please. I think I didn't adjust the light properly on my face, but anyway.
मैम जो फाइलोजेनेटिक ट्री होता है उसमें मैक्सिमम लाइक्लीहुड और एक मैक्सिमम पार्सिमनी होता है, मैम उसका डिफरेंस एक बार बता दीजिए।
उसका difference, right? So what we do is, again, I will share my screen. Right. So that is one thing I tell you here, is that is absolutely computational thing, meaning that is for computer people mainly. Okay, so you don't have to worry too much about that if you don't understand some part of it. All right. And it's not here, I have not written it here, but again I can share that video with you where I had talked in details the maximum parsimony. And when you do this in PhyLab, if you remember, and in the MAGA, I taught you this in the class. So what do you want from me? You want short note on this? I can send it to you quickly. What else? Do you want me to tell some important questions? I don't listen. I don't hear from you. That's the thing.
Yes, ma'am.
Keep your mic on. Right. So let's start unit wise. Where is your syllabus? Is here. Right. Sorry, it took me some time to open the syllabus. So all of you are with your syllabus, and what we are trying to do now is to look for some important questions, right? In this syllabus on bioinformatics. So one is introduction to bioinformatics, and here I expect you to know the contribution of who is the founder, pioneer in bioinformatics. I taught you this in the class. What is her name?
I'm Dr. Margaret Dayhoff.
Yes, you should know her contribution. That becomes the history, and I can share a table with you where all the sequential development in bioinformatics is written. So I think, can we share these documents here on I think so, file, and I don't know my computer, right? Or maybe I share the screen and then you can know. This is one thing, her contribution. Sorry, I was about to put this meeting off. So let's do it quickly. Why is that? This is one thing. Then another is, then this genomics, transcriptomics, proteomics. Will you be able to answer? Transcriptomics, genomics, metabolomics. Will you be able to answer these questions?
Yes, ma'am.
With the examples? Because any one of this, I'm sure it's going to come in. It's going to come in right short notes. And then writing the definition is fine, but you should also give an example to explain these things. I think you'll be able to do it. Is it all right? These are the important segments for defining and short notes. Metabolomics. And what about system biology? System biology. You know what is system biology? System biology, anyone wants to tell me what is system biology?
Ma'am, it is the integrated form of all omics layers.
You know, rather than defining it this way, if you say system is what? When we talk about a system, it may be a human being, it may be Arabidopsis thaliana, it may be maize. So when we treat this as one system, and here the computational biologists, the mathematicians, the biologists all get all information about, they cooperate and collaborate with each other to get information about one system. This we call as system biology, meaning they collaborate and work together, but their reason for collaboration is one system, meaning maybe human being, maybe like the example of system biology is what? Human Genome Project, where human was a system and all its genome was deciphered. Apart from deciphering the genome, then the statisticians and mathematics and genetics people, they sat together to work out all the genes. Then the metabolics or chemistry people, they come together to find out all the metabolites associated with this. And then computational biology and everyone, but the unit was what? Humans. All right, now you'll be able to write this short note on this on your own. I don't see anything difficult in this. No theoretical, no rectification, straight answer it like this. Is it all right?
Yes, ma'am.
Yeah. Next is biological databases. What are biological databases? That you'll be able to answer. What are biological? I taught you this in the class also. What are biological databases? No, you don't have any problem with the biological databases, so I quickly tell you what is going to come in the exam. Is GenBank maybe, PubMed, short note. What is PubMed? All of you know, PubChem. And this new term has come, Plant PepDB. You know about this? I didn't teach you this in the class. What is this Plant PepDB? Anyone? See, you people, I don't know, day after is your exam. And if I ask you like this online, I think you can Google it and then you can tell me on, if you don't want to speak on the this comment section. But I don't know, there are 7 participants here and what they want to do, I don't know.
Hello.
Hello, Hanji, बताइए, just tell me now.
It is a plant. Ma'am, it is a plant specified databases.
No, I tell you what, you missed the main term. Plant specified peptides. PEP, say you remember the peptides. It's a database, and what is good thing about this? It's going to come in the exam. What is good thing about this is that it is developed by Indians, that is NBPGR people have built this, right? And this is very, very useful because we use these peptides in medicines. जो आप medicine खाते हैं आपको, तो you don't know what is the chemistry of that. Sometimes it is some chemical, at other times it is peptides maybe. So this is extremely useful in drug designing, medicines, and other things. So if you are asked in the exam to write this database, what all you will write is, you don't forget this peptide word here. That is the most important thing here, right? So this is plant peptide database. This is clear from its terms itself. And because plant is a source of several peptides which are used in medicine, right? So when you have gathered all of them together at one place, it is of immense use, right? And then apart from this, good thing about this is developed by a team in NBPGR. That is why we have a special interest in this. So once you know these, you try to remember these few small things, and that is, then you don't have to cram up things, right? Now you'll be able to write few lines at least about this plant PepDB or not. Same was for system biology. No rectification, just understand things and then write. At very few places, I expect you to write the figures, meaning figure of what? Say for example, I don't expect you to write that NCBI has how many sequences today present in their database. I don't expect this. So no cramming, but you should be able to tell what is NCBI and what good things it has been doing, right? Okay.
Yes, ma'am.
Anything else you want to ask? Let me go further. So, UniProt, NCBI, and you will be able to write. Primary, secondary, this PubChem also is newly added to the syllabus, and that is why it comes in the exam. What is PubChem? PubChem.
Ma'am, it is a chemical database. It contains information about the chemical structures के बारे में बताता है और उनकी
Right.
bioactivity के बारे में, toxicity के बारे में.
Right. And this is hosted at NCBI. These small informations, if you give, you will get full marks. Right? Where is it lying? It is at NCBI. And you can also cite one example for which it has been very useful. All right?
Ma'am, example can be PubChem Sketcher and PubChem BioSA.
Yes. Meaning, go a bit more in details about that. Okay?
Okay, ma'am.
Meaning, like I told you, for system biology, you can write the example of human genome project. So here also explain this a bit more in details. And another thing which I observed recently, which is going to come in the exam, is primary and secondary databases. What are those?
Ma'am, primary database has raw data and we can say secondary have annotated and curated data.
And composite.
Primary
See, again, I tell you what is happening is that while studying, you don't focus on the terms. Primary is derived from original research. Got my point? You didn't say
Yes.
these words. Primary is derived from, even if you will write the answer and if you're missing the exact terms, they don't require any cramming, right? Primary is derived from original research. Sequences which are coming from original research. All right? And secondary is coming from the primary databases or curated databases from the original sources. Please write these differences. Okay? Primary database,
Yes,
you can write the example, and you can write the example of a secondary database. Those sequence, the gene identification databases, most of them are secondary databases, right?
Ma'am, UniProt, second.
UniProt, protein, that's a protein database, that's a primary database. But the secondary databases are maybe any particular, this peptide database is a secondary database. Plant PDB is an example of secondary database because it has taken out all the sequences which have been derived from plants. Whomsoever sequenced that, those sequences they are not bothered, but they talk about the sequences which have been which refer to peptides in the protein. That becomes the example of secondary database, right? Now, what is this composite database?
Ma'am, it is the collection of several primary databases resources.
Several, and it has a purpose. It makes them one unit. There should be a database, should have a purpose, you know. And the composite database, for example, you can write with the help of example. Like there is a database on maize, maybe maize or any organism. And if you open that database, you will see that it not only gives about the complete genome information, genes information about that database, it also gives all the proteins, it also gives all the chemicals, it will give you all the chemical pathway and everything. All right. So what is that? That's a composite database. Because it is not only talking about in fact, references also, it will give you all the new research on that. So PubMed is on original bibliographic database. GenBank is original sequence database. Whatever sequences people get, they load them on GenBank. But MazeDB is a database which is a composite database. It has all information about maize. And secondary database, I gave you the example of plant PepDB. You can write that example, that you don't have to cram up many things. Does that work for you? Is it all right? Right?
Yes.
Don't grab things, just try to grab the crux of that. What is the main thing here, right? Then only you learn. Blast bhi ho gaya, PubMed you now know, and PDB you will be able to write the difference between NCBI and GenBank and PDB. You will be able to write. What is the difference between these two? Quickly tell me, then I go to sequence alignment.
Yes, ma'am.
Ma'am, GenBank is a nucleotide sequence retrieval, and PDB, ma'am, tertiary,
So,
मतलब
one
3D
thing,
structure बताता है
Angel,
protein.
why do you want to add retrieval here? It's a database which contains all these sequences, right? DNA or
Yes,
RNA,
ma'am.
nucleotide sequences. That's it. No retrieval is needed. Okay? Right? And what about the other one?
Yes, ma'am. मैम पीडीबी मैम 3D स्ट्रक्चर बताता है प्रोटीन का, उसको
And that,
विजुअलाइज भी कर सकते हैं.
right. Uski unit kya hai? What is the basic unit on the basis of which it is built is?
अमीनो एसिड.
No. That is why, you have to be clear about things. That is based on the crystal structure. That particular protein in PDB, like you are a chemistry students, what they do is first they crystallize that protein, purify, crystallize. And after crystallizing, they have its 3D structure by X-ray crystallography. So that way it tells you every atom at which position, which atom is there, right? And that is how you get a 3D structure of the proteins. So the basic unit in PDB database is X-ray crystallography data. And in UniProt, what it is? It is the protein sequence, peptide sequences. Got it? You can discuss
Yes,
with
ma'am.
me anytime, and please, if you want to go ahead in life, you should be clear. No cramming, no ratta type of words, but you should know what you're talking. Okay? Let's go ahead. And another question. So do I go to the next unit now? Next unit? Okay? Still, you can anytime ask me questions on WhatsApp. Send me the questions, I'll be able to answer. And I'll take you what exactly is expected from you. Clear answer. Right? Now let's come to sequence alignment. And in sequence alignment, I taught you this in the class also, I remember. And the most important part of this is difference between similarity and homology. You know what is the difference? I think I might have 9 minutes remaining. What is the difference between similarity and homology? Anyone? What is similarity? What is homology? What is identity? First topic written in the syllabus, and it is going to come in the exam in any form, I tell you. Long question, short question, or whatever. And you have to answer this with the help of an example. And similar type of question is orthologous and paralogous genes. This is going to come in the exam, I am telling you. Homology and similarity, orthologous and paralogous genes. These two things will be there. One of these will be there in the exam. How will you do that? अभी आपके युवा people are not ready for the exam, I think so.
मैम जो होमोलॉजी होती है उसमें कॉमन एंसेस्टर होता है, और जो सिमिलैरिटीज होती है उसमें विदाउट हैविंग अ कॉमन एंसेस्टर हम कह सकते हैं कि कुछ सिमिलैरिटी मतलब कुछ सीक्वेंस सेम होते हैं.
Right. So, आप इसको clear, you can talk about this clearly, but you are expected to write the examples of similarity and homology. So, the clear difference in these two is that in homology we talk about evolution, phylogeny, right? But in similarity we don't talk about. It's a mathematical similarity, how much similar the two sequences are. Okay? You got it? Homology is derived on the basis of similarity by making a phylogenetic tree, but similarity just refers to point-wise point matching of the nucleotides. Is it clear now? I hope people don't believe in cramming things. You try to understand what we are talking there. And what about orthologous genes and paralogous genes? They are going to come in the exam, I am telling you. What is the difference between orthologous and paralogous genes? Google it, at least it will come out of your mouth. What is the difference between these two things?
मैम ऑर्थोलोगस सीक्वेंसेस वो होते हैं जो हमारे सिमिलर एंसेस्टर्स, मतलब जिनका सिमिलर एंसेस्टर्स होते हैं, और पैरालॉगस वो होते हैं जो जीन डुप्लीकेशन इवेंट से सेपरेटेड हुए होते हैं।
राइट। एक और था टर्म यहाँ पे, पैरालॉगस, एनालॉगस भी होता है एक शायद, है ना? Just 1 minute. You must please, you answered it, but I could make out from your answer that you don't know it absolutely. Right? Paralogous. Logous, and there is one more, and homologous. And homologous. पैरालोगस वर्सेस ऑर्थोलोगस आपने मेरे को बता दिया है, बट यू हैव टू राइट द एग्जाम्पल हियर ऑल्सो. You remember the example of fur in the birds? All the birds have feathers. That example comes here. So I think if you people want to score good in the exam, you should prepare an answer for this also. It's going to come in the exam. Write a short note on orthologous and paralogous genes. And maximum likelihood, distance neighboring, and this maximum parsimony, I'm sending you the notes for this, and I will do it for orthologous, paralogous, and analogous genes also. I'll send you notes because it's going to come in the exam, I'm telling you. It's a very important topic. Now you ask me if you want to ask anything, because I have 8 minutes remaining.
Ma'am, maximum likelihood.
Sorry, you were not audible. Can you speak it again?
Ma'am, what is maximum likelihood?
This is what I said. I'll send you notes for this. This is absolutely computational terms, and I will do it I'll make a small paragraph point wise, so that if it comes in the exam, you are able to answer. I'll just do it in half an hour after this and send it to you. And same is for orthologous, paralogous, and analogous genes, because that will also come in the exam. And then you will write theoretical things. No need to write theoretical things. Just write it from there. I'll write it point wise for you, both these answers, right? And then I can meet you again if you still want to discuss anything. These 2 notes I'm sending. What you do is, today is 31st. So I wish you Happy New Year. You're in India, the New Year is about to start, right?
Yes,
ma'am. Happy
After
New Year,
1
ma'am.
or 2 hours. So that's so great. So I wish you Happy New Year, all of you. And what you do is enjoy your upcoming New Year. I'm sending you answers for 2 questions. And if you still have anything to ask, you are most welcome to have a class again, or you can send me the questions. I'll prepare the answer for those and send it to you, right? And I also invited you to join me in a project next semester, starting here from 13th of January. And there I'm teaching microbial community analysis to the students here, and maybe I can teach you also. That becomes your project if you have in your microbiology paper. Otherwise also, after having studied bioinformatics, it will make you a very, what has happened with me, like I can get a job here very quickly. Why? Because my specialization is bioinformatics and microbiology. Meaning I see a lot of offers around, but I don't know what I'm going to do. But that's a very, very upcoming subject, and you must train yourself in that. All right? Anything else if you want to ask?
मैम एक बार गैप्स एंड पेनल्टी वाला थोड़ा सा.
Right. Gap is what? During multiple sequence alignment, right? During multiple sequence alignment or pairwise sequence alignment, right? What we do is we keep one nucleotide sequence below the other, right? And there are 2 types of alignment. One is optimal alignment, one is local alignment, right? In optimal alignment, what is our focus is to get the best alignment. The best alignment, meaning for every match we give score of 1 plus, and every mismatch we give a score of -1. All right? And then we calculate the net score for optimal alignment. All right? Sometimes to increase our score, we insert the gap, and with insertion of the gap, there are more matches. All right with this? And our score increases. Done? Now, but we cannot insert gaps just like that. We have to have a penalty for gap. So what was your question again? What is the difference, or what was that?
Ma'am, yes, gaps and penalty difference.
Penalty, right? So to get optimal or a good alignment score, what we do is we insert gap. And you can explain this thing with the figure. If you remember in the class, I prepared a figure. Draw one sequence above the other. If you have not
Yes, ma'am.
remember it, go to that video and watch it. And you have to show in your answer that you know this, right? And then again make that sequence and insert the gaps. And then tell that the gap has a penalty, while in the matrix, when we want to calculate the score, this is what penalty is. Then also explain what? Now there's a lot of sun here. I need to change. Maybe the meeting is about to be over. What was that? And if you remember, I told you about these scoring matrices. Day-off matrix, blossom matrix, if you remember from the class. And what are these matrices? They, on the basis of their experience, tell you how much should be the penalty, right? On the basis of their studies and this thing. So for penalty, we have different matrices which tells us how much penalty is to be given if we insert a gap in these particular sequences. So you can follow these sequence alignment algorithms. They use any of these matrices to give the penalty. I hope you got it now. Still, I offer you, if you want to write your answers and share with me, I can correct them, make them best wherever anything is to be inserted. But please help yourself. Don't feel. Otherwise, you'll be nowhere. The time has come. The skill and the knowledge matters. Nothing else is being bothered. And if you are not going to study and prepare yourself for conversation, for interviews, and for actual knowledge, you'll be nowhere. Anything else you want to ask? I just have 2 more minutes. And before this meeting is ending abruptly, I think it's a good idea to close this now. If you have any questions, quickly ask, or you can always ask me to meet again anytime. But my purpose is that your exam should go well. And you should prepare yourself for the next semester, for which, your first semester, which paper you took? DSE. What was that?
Plant diversity and systematics.
No.
Mathematics.
This is your first DSE, I suppose. First.
Yes, ma'am. DSE.
Perfect. So learn things well, and only in exam students study. So learn well so that next semester in microbiology, we can do a project on microbiome diversity analysis. That will include both your statistics knowledge, bioinformatics knowledge, and a good genomics knowledge. Right? So you want to ask anything else? Prepare yourself well. I find you people are not prepared till now. You have an exam day after tomorrow, but you are not ready for this. All right, this is what I can do. I cannot study instead of you. So best of luck, and I wish you Happy New Year again, and enjoy your
Thank
day
you, ma'am.
and study well, right? Welcome. And I want to disconnect it now. Okay, best wishes to all of you for your exam. Bye.